Assalamu alaikum. I, Dr. Mohammad Akram, Department of Eastern Medicine, Government College, University, Faisalabad. Digestion and absorptions of carbohydrate. Major dietary carbohydrate are starch, sucrose, and lactose. Starch is a polysaccharide, while sucrose and lactose are disaccharides. Digestion in the mouth. Salivary alpha amylase Tyline cleaves alpha 1,4 linkage of starch and converts it into dextrins. Digestion in the intestine. Pancreatic enzyme. Pancreatic alpha amylase cleaves alpha 1,4 linkage of dextrin and converts it into disaccharides, maltose, trisaccharides, maltotriose, and oligosaccharides containing alpha 1,4 and alpha 1,6 linkage. Enzymes of intestinal epithelial cells. Maltase. It cleaves alpha 1,4 linkage of maltose, releasing two glucose molecules. It also cleaves alpha 1,4 linkage of oligosaccharides and releases glucose molecule. Alpha dextrinase cleaves alpha 1,6 linkage of dextrin, releasing glucose molecule. Sucrase cleaves sucrose and releases one molecule of glucose and fructose. Lactase cleaves lactose and releases one molecule of glucose and galactose. Clinical notes: carbohydrate malabsorptions, isomaltase sucrase deficiency. It is a deficiency of sucrase. Patient cannot digest sucrose found in Eskimos and North Americans. Treatment: avoid dietary sucrose. Lactose intolerance. It is a deficiency of lactase. Patient cannot digest lactose, thus unable to drink or eat dietary products. Lactose intolerance. Metabolism of lactase deficiency is not clear. It can be age dependent in older people, acquired or temporary, patient with diarrhea or congenital deficiency of lactase since birth. Particularly common among blacks and Asians. Symptoms: bloating, abdominal cramps, and osmotic diarrhea 2 to 4 hours after eating dairy products. Treatment: avoid milk or dairy products or add lactase pills to eat diet. Cellulose and indigestible polysaccharide. Cellulose is a fibrous dietary polysaccharide containing beta 1,4 glycosidic linkage between glucose residues, which cannot be digested by human due to the absence of its enzyme cellulase beta 1,4 endoglycosidase. Absorptions of carbohydrates. The final product of carbohydrate digestions are glucose, fructose, and galactose. These sugars are absorbed by intestinal epithelial cells, carrier moving against the concentration gradient, transport all of them into the cells. It transport protein that carries sodium ions also transports glucose, and it requires sodium potassium ATPase, which forms potassium, which pumps sodium into the blood along with glucose against the concentration gradient. Digestion and absorption of protein. Protein are digested to amino acids. Gastric enzymes, pepsin and pancreatic enzymes, trypsin, chymotrypsin, elastase, and deoxypolipeptidase convert protein into oligopeptides and amino acids. Enzymes of intestinal epithelial cells, aminopolypeptidase, dipeptidase, and tripeptidase convert peptides into amino acid. Amino acids are absorbed through intestinal epithelial cells and enter blood. Digestion in the stomach. Gastric enzymes. Pepsin is secreted by the chief cells of stomach and breaks proteins, large polypeptide into smaller polypeptides. Digestion in the intestine. Pancreatic secretion includes bicarbonate and proteolytic enzymes. Bicarbonate neutralizes the stomach acid and raises the pH of the intestinal content. Proteolytic enzymes include endopeptidases and exopeptidases. Endopeptidases cleave the central peptide linkage of the protein chains. Examples are trypsin, chymotrypsin, and elastase. Exopeptidases break down amino acid at a time from the carboxyl end of the peptide carboxypolypeptidase. Enzymes of intestinal epithelial cells, proteases, exopeptidase. cleaves one amino acid at a time from the amino end of peptide, for example, amino polypeptidase. Dipeptidases and tripeptidases cleaves dipeptides and tripeptide and releases amino acid. Absorptions of amino acids. Amino acids are absorbed by intestinal epithelial cells. Two different types of transport mechanism carry amino acid from intestine into the blood. Sodium amino acid carrier system, it transports protein which requires sodium potassium ATPase activity, transport sodium ions and also amino acids. Gamma glutamyl cycle. It is a glutathione dependent process in which amino acids are transported across the cell membrane, attached to gamma glutamate and released into the cytoplasm. Digestion and absorption of lipids. Digestion. Major dietary lipid is triacylglycerol. Digestion takes place in the small intestines and requires bile salts and pancreatic enzymes. Bile salts emulsify the dietary lipids. Pancreatic lipase, along with colipase, converts triacylglycerol into 2-monoacylglycerol and free fatty acid, which are packaged into the micelles. Micelles are microdroplets of fat emulsified by bile salts, which also contain cholesterol and fat soluble vitamins. Absorptions. Micelles get attached to the microvilli of intestinal epithelial cells, which absorb 2-monoacylglycerol, free fatty acid, cholesterol, and fat soluble vitamins. Resynthesis of triacylglycerol. In the epithelial cells, fatty acid from micelles react with 2-monoacylglycerol to resynthesize triacylglycerol. Triacylglycerol is released in the lymph as a component of chylomicron, which then enters blood. Clinical notes: lipid malabsorptions. Loss of lipids, including fat soluble vitamins A, D, E, and K, and essential fatty acids in the feces, is known as steatorrhea. Steatorrhea can be caused by a number of conditions related to the deficiency of bile and pancreatic enzymes or defect in the absorptive cells of the intestine. Thank you very much.